What I did today was I was here for Matthew's defense yesterday. That went very well, so congratulations to Matthew. And thank you for having me here for this opportunity. I handpicked a few experiments that I had over the last 10 years or so regarding protists, primarily because the thesis yesterday was on protists, so that's why I picked this theme. And so I'll give you an overview of some of the things we've done and some of the progress we've done trying to figure out how to work with protists in soils. Many of the techniques that we were using were outdated and really not adequate for even getting them out or seeing them properly, yet alone counting them and looking at the diversity. And so we tried to introduce a lot of more modern techniques into how we deal with these guys. So I'm going to bring you down into my world and kind of my headspace, where this is where we work and how we think about the world. It's has a lot of mineral particles, and there's a lot of organic matter coating the mineral particles. We're dealing with water films and pores that may or may not have water, and it's a very dynamic environment. It changes every few hours. The temperature changes, the moisture changes, the pores get filled and emptied with water. And what really dominates this sort of system, if you have roots in it, is the plants and their roots, because they're really sucking up all the water and many of the nutrients. And so the bugs in the ground, their job, from my perspective, is to break down the organic matter and put root-available nutrients into the soil solution. And that's how we're looking at this system. Now, the problem with this is that it's dark, and everything's stuck to everything, and you don't see much at the microscope, and the chemistry becomes very complicated because everything's stuck together. And the very fact that you're sampling is destroying all of the structure that there is, and so you've got to do a lot of reconstruction, and that's where most of our errors creep in. So this is an example looking at cows in a pasture. It gives you an indication of what we have here is the above-ground producer subsystem, which is what most of ecologists study. And then we have the below-ground decomposer subsystem, which very few people study. But the two are interlinked in many ways, because the stuff above influences what happens below, because there's a return of nutrients. In addition, there's always some sort of man-made management. And overall, you can look at the CO2 going into the green stuff, and eventually the CO2 is decomposed back up into the air, and that's kind of the cycle that you're looking at. And in the middle, there's a lot of biology that happens. So how do we simplify this and look at it in different ways? We can put a little cartoon here, and you can look at how things are interacting. It gets fairly complicated because you can have a nematode that's feeding on fungal hyphae in organic matter. You can have some mite that comes along and eats the nematodes, and the fecal pellets come out, but they get colonized by fungi that break that down. You can have the nematode fecal material getting colonized by bacteria and protists, and the bacteria and protists, the protists feed on the bacteria, and then you can have a bigger one that comes along and gobbles them up, and then you can have another one that feeds on them, and then you have all of these complicated interactions where the organisms are living in their food and excreting material that becomes food for other organisms that are living there. So it's all, both the food and the excreted material are mixed, and that is that looking at that sequence of decomposition is interesting. But you can simplify it and structure it so that we can look at the primary saprotrophs, which are the species that are dumping enzymes into the environment to break it down. And we talk about the secondary saprotrophs, which are the species that are eating those. And we can put those into a variety of categories. I have some here. The fungivores feed on the fungi, and the bacterivores feed on bacteria. But we also have the cytotrophs that are feeding on the protists. We have the tridivores that are ingesting organic matter. And we have the osmotrophs that are only taking up the nutrient solution, a lot of yeasts and some bacteria and so on. And then we have other consumers, which when we get into the predatory mites, for example, some of the larvae, some of the spiders, and so on. And we can make these more complicated. This is an example from Moore and de Ruyter. But one of the lessons from the last 20, 30 years really is that we're looking at, there are two main parts to this food web, and three if you want to look at the root feeders as well. But we tend to see models in manuscripts that look at this fungi-fungivorous pathway and the root and root feeder pathway. And then the other main component are the bacterivore pathway. And these are linked near the top by the consumers that are feeding on the secondary consumers that are feeding on the primary consumers. And so the experiments that I'm going to talk to you about and a lot of the data that I'm going to show you really deals with the bottom bacterivore pathway, asking the question, what is the role of the protists in this system and do they really matter? Historically, we've tended to think they don't matter because their biomass is negligible and their numbers are very low. It turns out that their numbers are very high. We just didn't know how to look at them. And it turns out, lessons from ocean microbial ecology, that the biomass is irrelevant. It's what they do that matters. It's about how fast they're turning over nutrients. So if we simplify biology, you've got bugs eating stuff, and there are ways we can measure that. The output is these four components. There's a heat output that we measure in joules. There's waste gases, liquids, and solids. Every species does that, right? The gas is always the respired material. Eukaryotes, it's CO2. Waste liquid is usually to get rid of waste nitrogen. For protists, it's ammonia. A lot of invertebrates, it's ammonia. And then there's the solid parts, which is the undigested material. It will include a lot of the cell wall material, the cuticles, and things that are not so easily digestible. So let's have a look at what we're dealing with in terms of protists. The reason why the literature shifted from using words like protozoa in the 70s is because it was no longer possible to distinguish between what is and isn't protozoa or algae because they're all intermingled. And this is an example looking at diversity of eukaryotes. You can see that these are the protists. The animals are up here where it says metazoa, and they belong to the opisthokonts. And the plants are a branch out of these chloroplastida, the green algae. And the rest are all the unicellular eukaryotes. And the algae occur throughout in various places. And you have some like the apicomplexa up here, which are animal parasites, but they used to be algae. And so they have a remnant of a plastid that is still useful in the infection process, but they haven't been photosynthetic for a very long time. And so the term protist is used to describe these for that reason, and that happened in the 70s and 80s when all the societies and journals changed their names. And the ones that occur in the soil tend to be the amoebozoa. We have obviously the fungi that dominate the decomposition. We have the Cercozoa, which are most of the little flagellates you encounter. And then we have some ciliates that are always present, but inactive most of the time. But when they're active, they really dominate the system. And then we have a few things here in the excavates, which are much less important to the overall food web, but they're very interesting organisms. And this is a new paper we have coming out that really looks at and revises the classification of the protists, and we have here a much better understanding of what the diversity is and how they're distributed and so on. And we're looking at several kingdoms and about 50 phyla. There's quite a lot of diversity, which was really unknown, especially the soil diversity was almost completely unknown until early 1990s, so fairly recently. And the ones that were missing from the system were these guys, the Cercozoa, and they have filopodia. These are little flagellates that spread thin and flat on substrates, substances, and they extend these sub-micron pseudopods that branch into the substrate. And these you don't see because they're transparent. They're less than 5 microns thick, and they're hugging whatever particle they're in. But they're also in the material and covering most of the surfaces. And it turns out that a lot of the nematodes, when they're going along the surfaces and it looks like they're sucking on bacteria, they're actually ingesting a lot of these guys. So there are, this is a very large group, and there are many, many species. We're only beginning to have a sense of the diversity that there is, but there are many, many genera and new ones. Several new genera are described in manuscripts that are coming out on this group. And this is some photos from David Bass' papers showing you some of these really fine pseudopods that can extend into just about every nook and cranny that there is in the soil. And they get their bacteria there. They don't have to be able to get into the pore to extend a pseudopod to get the bacteria there. So there really is no place for the bacteria to hide. And these ones they're flagellates. And so when they're let go of the surface, they round up and look like a regular flagellate, and they swim off and they reattach to another surface. So there's some mobility there, and they can explore their space. So I'm going to talk a little bit about techniques, and then I'll talk about function, and then I'll try to talk about how we can measure the impact of these guys in the ground. So what we did late 90s, early 2000s was to relook at all the procedures and adapt them and come up with new ones so that we could start looking at these. And the main thing really was to encourage soil scientists to abandon the MPN procedure, because all of the other microbiologists had, but for some reason it had lingered in the soil science community, even though microbiologists were not using it anymore. And so we had to come up with new procedures to get the protists out and counted. And these relied on microscopy and modern techniques. And making better use of the direct count methods. To do that, we had to come up with statistical procedures that were useful. And so we looked at existing statistical procedures. And the ones that work the best are the ones that are used by bird people for counting birds and estimating bird numbers. The ones that are looking at large mammals. And a lot of these have to do with transect techniques, whether it's by walking in the forest or by flying over an area. And those statistical procedures work very well at the microscope. Other things that needed to be done was to standardize the media. The way people were preparing media, if they used them at all, was inappropriate for protists, and there was a lot of knowledge transfer here in developing new standard soil solutions and culture media that were appropriate for soil protists. We had to come up with extraction procedures, and so we have different extraction procedures depending on how the organisms move. The ciliates need a separate technique, the amoebae need a separate technique, and the flagellates need a separate technique. And then we had to try to grapple with the functional groups and try to understand why all of these thousands of species that are eating bacteria are different. Can we not just have 3 or 4 species eating bacteria if that's all they're doing? And I'll show you an example of why it is that they're not all the same and why they're different. And as the molecular techniques, especially the environmental genomic stuff, was coming on board, it became easier and easier to have a look at these, at the diversity, and to ask better questions. So some of the non-culture-based protist extractions are Petri plate-based, very simple to do, and microscopy counting techniques. I won't go into those. We were able to apply those to look at soil community structure. We were able to show that we could follow dynamics of species abundances and composition in a great deal of detail. And we could almost follow this hour by hour on the field and see how they were changing. We know that the abundances change through the day and through the night, and depending on when you sample, you will have high or low abundances. It changes through the seasons, and the species change through the weeks as the seasons progress, and depending on when in the year you go sampling, you'll have completely different species present. If you go on a dry day, you'll have some kinds of amoebae. If you go on a wetter day, you'll have completely different kinds of amoebae. And so we were able to identify some of these basic parameters that were important in shaping the community, but also, more importantly, getting a handle on how fast the community was changing. And it's quite fast. And then there are all the obvious things that matter, including soil physical property, the details of the organic matter chemistry, the soil solution chemistry, and the way the field is managed, and what kind of plant management and field management you have. All of those mattered. And we've shown those particularly for pastures, where we had a big grant. So looking at function, optimal foraging, for those of you who know your ecology, is where we draw most of our concepts for this. What we know is that the different species are very picky about what bacteria you give them. You cannot grow any species on any bacteria. You have to spend some time getting the right combination. And even these little flagellates that are ingesting one bacteria at a time will spend a lot of time tasting and choosing their bacteria. They'll find a microcolony, they'll begin to touch it, and then they'll begin to ingest one, and if it's not good, then dump it out, and they go and try somewhere else, and they just keep tasting until they get one that they can ingest, and then they'll digest it. And so this tasting you can see at a microscope, and you can measure it and take pictures and stuff. And it's now quite well known, and you can do molecular biology to show that there's a lot of mixed bacteria culture. There's a lot of selection of grazing, but different protists that happens. From optimal foraging theory, again, you have these numbers that you can measure. They tend to be used more with predator-prey interactions, but here you're looking at things looking for bacteria. And it works the same way. So search time and capture efficiency and growth rates and so on. And we can measure all of these things at the microscope fairly quickly. And ecologists have something they call a functional response curve. I've used here terminology that is used in chemistry because you're more familiar with it, and it's easier to explain it through those. But it's pretty much, what you're looking at is pretty much an enzyme reaction, where you have a maximum rate and a minimum rate of ingestion, and you have a Km that you can calculate, which is a half Vmax value. Sometimes this is shaped like an S, and sometimes it's linear. Well, very rarely it's linear, but sometimes it's shaped like an exponential increase. The more interesting ones are these S-shaped ones, and they tend to, I think, describe most of the encounters that I've looked at. And an example with one combination, this is a ciliate called Cercozoa, which is very common in the soil, and we've looked at it feeding on bacteria, on Chlamydomonas, something a Cercozoa, and then something a bit bigger, which is a colpodid, which is a small ciliate. These are old graphs, so don't worry too much about how they look. I didn't want to spend too much time fixing them up. But the main point here is to look at the colors, and you have the 4 degree, 27 degree, 17 degree in this particular order, and it shows you how fast this bug can feed on bacteria. But then when we look at something else, the pattern changes. On the cercomonad, it hardly feeds on them at all at 4 degrees or at 17, but at 27 it seems to do quite well at the higher concentrations. If we look at this feeding on Chlamydomonas, which is a completely different organism, you can see that it does quite well at 17 and 27, but not so well at 4 degrees. But at 4 degrees, the chlamydomonas is a better food option at the lower temperature. And so depending on the temperature and what bugs are present, they're going to be feeding on different things. And this is just one example with this particular ciliate. And when we look at it with Colpoda, this is kind of at the limit of how big of a thing it can ingest. And you can see that at 27 degrees it's more effective than at the other temperatures doing this. And we can calculate things like growth efficiency on the various food at different temperatures. And looking at this, you can see that the 17-degree Cercozoa over here is the best combination. And for this is not really surprising because the soil there tends to be around 10, 20 degrees most of the year, 10, 25 degrees. And some of the other ones look like they're really not efficient. They'll feed on these things under some conditions if they have absolutely nothing else to feed on. And it'll keep them going and it'll prevent them from forming a cyst, but it's really not the preferred things. So these are interesting ways of looking at what they do and how they work. I've been doing this for a lot of species now, and I've been accumulating this data for about 10 years. And so if I remove the data and just show you where they tend to cluster, I have the Km value and I have the minimum bacteria concentration, which really helps picture this. You can see where the Cercozoa are feeding. The black circle shows the 17-degree, and the dots show you the 27-degree experiments. And you can see where these different kinds of organisms are clustering, where you have the Cercozoa kind of feeding better at these KMs, and they can survive at much better, at much lower bacteria concentrations. Some of the ciliates that are bigger require a much higher minimum bacteria concentration before they're active, but their impact on the community is much higher because they're ingesting much faster the bacteria, much higher rates of ingestion. And I spent some time trying to figure out why there was a gap here. And based on some experiments that I had a master's student do, it looks like that's where the nematodes fit. So the nematode bacterivore seems to start just about here and go up. So they have a higher minimum bacteria concentration, and their Km values seem to be roughly about here. I didn't push these experiments too far because that's when we started encountering the nematode feeding on the protist interaction. And I had to rethink what the nematodes were doing, and we had to do some radioactive labeling experiments to sort out whether they were actually ingesting the protist or if they were injecting the bacteria, and things became a bit complicated. So if we take a look at this and have a look at the impact these things may have, we tried to do at first radioactive labeling studies where we were labeling bacteria, various kinds of protists, and litter, and fungi, and trying to see where the label would end up. And we managed to show that here in the middle I have a amoeba that has silver grains in it, and we can get bacterivorous nematodes to feed on these guys, and some of the species tend to preferentially feed on the protists because they do better on those than if they're given only bacteria. And I think in reality they're feeding on the bacteria and protists together. It's hard to not suck them all up at the same time. And we looked at the fungivorous nematodes feeding on the fungi using this technique where I use the combination of calcofluor white for the cell wall and DAPI for the nucleus. And then you're looking at the nematode poking its stylet into the fungal hyphae and sucking out the cytoplasm. And what you see at the microscope is the nuclei are disappearing, and so you end up with empty cell wall. And by counting the number of nuclei per meter, or nuclei per centimeter, whatever you want to do, then you can calculate how much cytoplasm is going from the fungus into the nematode. And you can do that with the fluorescent technique, and you can do it with the radioactivity technique. So we got some pretty nice numbers there for the nematode functional responses. The problem we ran into with the radioactivity stuff is that it's very messy. It's very hard to get nice pictures. It's very hard to get good numbers. There's always a big error bar. And so we switched to stable isotope approaches a few years ago. And so we developed techniques where we could specifically label the bacteria, and then specifically label the protists, and then specifically label the fungi, and then look at little microcosm experiments to try to see what the rate of ingestion was and what the rate of CO2 accumulation was. And also to look at intact soil columns where we would inject the stable isotope and then follow the path up in the different parts of the food web. So I'll give you an example of those. And these were done in Devon with colleagues at the Grassland Institute. So it's a branch of Rothamsted. And these were pastures, and we would get these soil columns, and you can label the bacteria so that most of their, almost all of their atoms of carbon and nitrogen are your stable isotope. And you can inject that into the soil column very slowly and evenly. And you let it grow for a while, a few hours or a week or 2 weeks. And then you extract all of the bugs out of it. So the nematodes come out, the invertebrates come out. You can do a Tallgren extraction. You can follow it with nematode extraction and Protist extraction and so on. And then you can look at the kind of data that you get. So we did this with the bacteria, so that the bacteria were labeled and injected into the soil column, and then we labeled the protists, and we put them in the soil column. And the fungal ones we had to do on petroplate for technical reasons. It's very hard to have intact fungi out of the soil because you just shred them. So we have to do it slightly differently. What we could show was in a pretty good amount of resolution how fast and where the bacterial label was going. And so you can see it in the organic matter. You can see it appearing in the different groups of organisms. And I won't talk about this too much because it's in the papers and it'll take a while to explain. But I think that the main point really is that you can trace and quantify the nutrient through the food web very accurately. And the purpose of the 13C in ecological studies is to try to understand what they're feeding on. And so the amount of delta 13C tells you roughly what they're feeding on. And the delta 15N is useful in telling you how long the food chain is and how far up the food chain your bug is. And so by hand sorting everything into little categories where you put all of the collembola of a particular family in a group, and then you take all of the mites of a particular functional group together, and you just break it down as much as you can, and we push the resolution of what the mass spec machine can do. We're really putting enough little sample as we possibly can. And then for the protist work, we ended up having to modify the injector and the way the gas goes through the mass spec in order to improve the resolution of the machines. So we're actually operating that instrument way below what the manufacturer is describing. So we had to fiddle with it a bit, but we were able to get very good numbers for the protists as well. So when you're injecting your stable isotope labeled protists into the soil column, you can follow over a matter of days where the label is ending up. And it ends up in a whole variety of interesting places. You can see how much ends up in the earthworms, in the spiders, and how much ends up in the aphid that's feeding on the plant, and how much ends up in the nematodes and all of that. So it was very quite good. And those were also, we also did comparisons with natural abundances in the field to see how much stable isotope there was in the various groups of organisms just naturally, and we needed to make those measurements to interpret our data. So if we look at the bacterium protozoa pathway, because this is the novel one where there wasn't much described, you can see the bacteria labeled here, and they end up in the protists. And if you label only the protists, you can see that label going into a variety of places. And if I get rid of the pictures and look at this here, the protozoa label ends up mostly in the nematodes, and some end up in the colembula. And I think that's probably indirect because the protists are on the surfaces that the colembula are ingesting. And they also end up in the mesostigmatic mites because of what they're eating, and that's where the protozoa label ends up. And then from the mesostigmatic mites, we have higher level consumers that are feeding on the nematodes on the colembula and the mites that are passing the stable isotope up into the rest of the food web. And so this is where the protozoa label goes. That had been a mystery all this time. Nobody knew who was feeding on the protozoa and how many they were and why they were there. So that's the path there. So what we're really looking at is the bacteria stable isotopes, the bacteria biomass being transferred into the colembula, the nematodes, the protozoa, and the oribatid mites, and these in turn being consumed by the rest of the food web that we understand, and the protozoa pathway is also there and quite significant. So to really understand the role of these, it really isn't the biomass that matters. The only thing that matters is grams per minute ingested per cell. And when you work backwards from those, you can do all the cool things that the marine ecologists have been doing for a long time. And these techniques allow you to get there and do that sort of descriptive work. So things that we're working on now, we're analyzing the fungivorous pathway. I have a postdoc who just finished, and we have all of the data. And we're assembling these. We are comparing the numbers that I have from the functional response curves to the data we've accumulated from the stable isotope procedures to see how far apart they are and if they're the same. Hopefully they'll be the same. We've been trying to calculate growth efficiencies from the Respired 13C experiments, and we have already encountered a number of instances where the numbers that are currently used in the models, and those numbers primarily come from the end of 70s, early 80s. Some of you might remember the IBP program, International Biosphere Program, where a lot of these early soil ecology kind of work and these numbers were accumulated. And they were done, a lot of them, those were done with radioactivity before that was heavily legislated. We couldn't do those experiments today. And those numbers, many of them need to be revised because they're not right, or they may have been right for where the experiment was done, but they're not generally applicable. And so there are some corrections that we have already identified. We have tested the limits of the mass spec instrument, so we've actually had to modify it, and that is also a paper that's coming out about how to do that. We have improved our resolution as a result, and we are now able to calculate the protist mineralization component of the soil food web, which we didn't know before. And the numbers seem to indicate that about 1 standing biomass of bacteria per day is consumed by the protists. It's actually quite a large number. So lastly, I want to talk about disturbance. So what I mean by disturbance is agriculture, tillage, but it also means forestry, forest harvesting, and it also means looking at mine sites and soil reclamation kind of experiments. So I'll give you one example, which is from Georgia. We're looking at fields that had been in no tillage for various periods of time. So it was a chronosequence study. And what they were doing was no tillage cotton. And this is BT cotton. And they were getting chicken litter from the chicken operations and putting it on the soil as a source of organic matter because these fields had been in cotton or peanut for a couple of centuries, and all there was left was sand. There was less than 1.5% organic matter. You're essentially walking on beach sand, no structure, your foot goes in and the tractor gets stuck. And when it's windy, it gets blown around all over the place, and when it's rainy, you have trenches in your field, and the farmers spent a lot of time going through their fields and putting the cotton sticks back straight so that they'll grow again. I mean, it really was a very bad situation, which is why they switched to no-till in this region. And if you look at the solid carbon, we're looking at two of the fields that were still under tillage have about 0.5% carbon in there, primarily sand. And they're still growing a crop out of it, not every year, but some years. And then the other ones had been in no tillage for 4 years, 8 years, 25 years, and the 90-year plot was a forest site adjacent to the 25-year plot. And when you look at the carbon, you can see that over this time period, for that sandy soil and under that climate, this is roughly the amount of organic matter you're looking at. So it never is very high to begin with. But what was interesting was what happens here. And this was the return of decomposition. As the bugs went back into the ground, the organic matter, the litter that they were applying, was beginning to get decomposed. So there was more of it in the spring, and there was a lot less of it in the fall. And that never happened with these sites here because there just wasn't enough of a decomposition food web there to do anything with it. This is something that a lot of the organic farmers are encountering. You don't see it here because this is a coniferous forest, so it was dropping needles all year round. And we looked at the diversity of nematodes and mites and protists, and the pattern is like this for all of those, where you have an increase in the number of species over time. The shift that happens that is significant is primarily between 8 years and 25 years, where you go from a lot of R-selected species to K-selected species. And that you don't see in this kind of graphs. If you're just looking at species diversity indices or number of species, you won't notice the change in the kind of species that are there. So the bugs that are present in these sites here are primarily R-selected species, and they have very different biology and ecology from the ones that you see in the 25-year site that are primarily K-selected species. And so we tried to see if there was more evidence of this in the literature, and it looks like from our review that in most of the little that there's published, what it tends to indicate is that somewhere between 15 years and 25 years, the community structure switches from being in recovery to being recovered. And so the time frame we're looking at for that to happen is not a few months or a few years, which is what most of the agriculture experiments had assumed, but we're actually looking at a much longer time period to have the soil food web be able to handle the organic matter that's in there. And so we did some experiments in the same, also in Georgia, same place, which was very simple. We had holes that were about 30 centimeters in diameter and 75 centimeters deep. And it was, okay, so what we did was we dug the soil out and we replaced it with sand. We dug the soil out and put it right back in. And we dug the soil out, autoclaved it so nothing was growing anymore. We verified nothing was growing, and then we put it back in. So that's your sand and soil and sterile treatment. The soil is just dug and returned, no other treatment. The other thing that we did was there's no further treatment, and then there was a litter amendment and no litter amendment. And each one of these holes was in triplicate, and it was randomized in a Latin square design. And then this experiment was done again at 2 other locations, 2 forest locations. And we followed it for about 3 years, sampling every 2 weeks. What we saw was, if you look at these here, the intact area outside of the holes had a diversity index. This is Shannon-Weaver diversity index more than 2. And for the grassland site, after 30 months, these holes had a diversity index of about 1.6 or much lower. So the sand plots, for example, had almost nothing in there. And this is just looking at the protists. So we're not looking at the mites and spiders and things, just the protists. With water coming down and the rainwater being able to diffuse through this site, which is not very big, you'd assume that the protists would have no problem getting from here to there and just spreading. But that is not what you see. They're clearly able to get in, but they're not surviving. And the reason for that, because you see this in the forest and you see this in the grassland, so there's something really wrong with the soil. And it really has to do with the soil structure and the details of the profile and what you've done to the bed, because that's their habitat. So we're really looking at the details of soil structure recovery and recovery of the profile. And that's what takes a long time. It's not about the bugs being able to get in and survive. They need to have their habitat recreated, and that takes a long time. Another example, we went and looked at a large coal mine in Czech Republic. And this is near Sokolov, and the mine is the Vintirov coal mine. And you can see it in the middle here. This is black stuff. That's the coal. And this area here is the mine. It's an open cast mine. They've been digging it since the 40s. And you can see the size of the mine because you can see the size of the towns around there. Chodov is a town. It's not a little village. Sokolov is also a town. And you can see these villages around here. And the mine is much, much bigger than the size of a town. Again, here you can see the mine. The hole is about 200 meters deep, and the coal seam is about 20 meters thick. And they're just following it and bulldozing their way through it. What is interesting is this part up here, this stuff here. And that's where all of the rubble has ended up. So you've got these hills that at one end are more than 40 years old, and at the other end they're fresh. And that gives us a chronal sequence, and it gives us places to do experiments. And what they've been doing is looking at how to get this pile of rubble turned into what might look like a park and remediate it. And there are many plantation experiments along this all over the place, and those have been studied extensively by my colleagues. And I went there to have a look at it to see what exactly I could find in terms of protists. And what there's a book chapter and a couple of papers coming out, I think fairly soon, that summarize a lot of this stuff. But the main conclusion really is what we had found before from a review of the literature, that at this particular site, it seems that the food web transition occurs at about 18-20 years. And everything before that is our selected species and few species. And then when you get to about 18 years, you have K-selected community structure and a completely different kind of food web structure. And that's where things seem to kick in and where the plants seem to be doing a lot better. So I'm going to wrap up here so we have time for questions. I have a very brief set of conclusions. We worked on new standardized procedures, and we described appropriate statistical subsampling procedures so that we can actually have numbers that were useful. And we shifted the focus from abundances and biomass towards diversity and looking at this from an ecological perspective, and spent some time looking at the functional responses and what their niche might be. We tried to come up with procedures, but primarily the stable isotope procedure, to quantify some of these nutrient transfers and identify the pathways in the food web. And we had also we did some work looking at the effect of the kind of profiles that we had, the litter chemistry and the impact of weather and chronosequence from a management perspective on the protist and on the soil food web in general. And we increasingly think that what really matters to their environment is the pet structure and what state that is and what's in it. So I'll leave it at that.